for the first event, we will be talking about the misuse of science and statistics, as you already know. And I'm gonna start. The structure of the event will be that our panelists, we're gonna be introducing themselves, and you have the chat box. Please introduce yourselves. Feel free to share with us any networking website that you usually use. I will be sharing our Scientistt website along with our Twitter and our LinkedIn account, so you can connect with us. And I will be starting with introducing our panelists. They will be introducing themselves. You can please start at any order. And I want to thank them so much for taking the time to be with us today.
Thank you so much. Shall I go first? So my name is Elisabeth Bik. I was born and raised in the Netherlands and did my PhD there in microbiology. And I moved to the United States 20 years ago and worked at Stanford for 15 years. Then I worked 2 years in industry at startup companies. And in 2019, I quit all of that. And now I'm a full-time science integrity consultant. So I specialize in science integrity and have reported about 4,000 or 5,000 papers, specifically for duplicated images, which is what I'm good in detecting.
Thank you, Dr. Elisabeth. Now, Dr. Julia.
Hi, my name is Julia Jerke. I'm from Zurich, from the University of Zurich. I just recently did my PhD, so I finished last year, and my PhD was about research integrity in general. So misconduct and publication bias in specific, and we investigated methods to capture highly sensitive behavior, such as misconduct in surveys, for instance. And just recently, our group conducted a huge study among scientists in Germany, Austria, and Switzerland, where we covered topics such as research culture, conflicts of authorship, publication bias, misconduct, and working conditions in science. And so right now we are about to analyze all this huge, massive data set. And yes, so this is research ethics is mostly our research topic.
So thank you so much for introducing yourselves and for joining us. I want to thank everyone also for being with us today. So the structure of the event will be that I will be asking our panelists questions, and they will be discussing the topics that I will be proposing. Feel free at any time to post any questions that you have. If we have the time at the end, we will have a Q&A session where the panelists will be answering your questions. We will also have running polls throughout the session for you to tell us about your opinions about specific topics related to what we're talking about. And now for the first question, or the first topic we'll be talking about, is I want to know from Dr. Elisabeth and Dr. Julia, how easily can data, images, and results be manipulated nowadays? So you can answer like in any order you feel free.
Julia, you want to go first?
Yeah, I might start. I can't tell anything about image manipulation because this is probably Elisabeth's main topic. But data manipulation, we conduct, or part of my research project in my PhD was investigating publication bias over time and how it changed over the last 60 years. And publication bias, I'm not sure whether any one of you knows what publication bias is, but it's one form of publication bias might be an unnatural clustering of data and data points just below the thresholds of significance. So you have an unnatural clustering of significant results in comparison to insignificant results. And what we saw, or what I found in my research, is that it didn't change. So we already saw publication bias like 60 years ago. And what we saw as a difference is that publication bias has been present at a 10% level, mainly in earlier decades. And now we have publication bias anywhere. So at a 10% level of significance, at a 5% level of significance. And I think it's mainly due to advanced methods and software tools, which make it very easy to conduct p-hacking and to conduct mild forms of manipulation. like outlier exclusions or model specifications that you conduct to somehow find something in your data. It's much easier to find results that you want to find nowadays, I would say. But still, we found publication bias already many decades ago. So this would be my first answer. It has been present before, but it's much easier nowadays, I would say.
Yeah, I agree with that. I also think that just the complexity of data has so increased in recent years. It was 25 years ago that I got my PhD, and if I think about what you know, how my PhD papers look like, they were very simple. They had one figure and one table, something like that. And nowadays papers are just so much more complex. So I agree that any bias and fraud has always happened, but it's just a little bit easier now because there's just so many more data points per paper, and it's so much easier to for images, in which sort of that is my specialty, to use Photoshop. In the olden days, we had to take our blots to the photographer and they would make a photo. Now you can just make your own artwork, and so there's fewer eyes on it, fewer people checking what you're doing, and it's easier to manipulate a photo, much easier than it was 20 or 40 years ago.
Yes, thank you. Now, after this comes the question of the role of the peer review and the reviewers in influencing the process or the manipulation of data and in detecting it. So what do you think is the role of peer review?
Do you want to continue, Elisabeth?
Oh, sure. So, I mean, I don't think it's a peer reviewer's role to detect fraud, but they definitely have a role in detecting things that don't make sense. So I would not expect the peer reviewer to, for example, detect a heavily photoshopped image unless it's so obvious that you can just ask yourself, why didn't the peer reviewer see that? But like if it's very sophisticated and well done, I would not expect the peer reviewer to catch that. But I do think that a peer reviewer should have just some common sense. Like I'm currently dealing with some papers where there were about breast cancer, and then 50% of the patients were male, and like those things should raise a red flag. And I personally think that that paper was completely made up. But yeah, you can clearly see that a peer reviewer has not really looked at that data and asked questions. And there's just many other cases where a peer reviewer should just spend a couple of extra seconds looking at the data. Is it too good to be true? Are the and just think about that it could be fraud. Like some papers will be fraud that you encounter as a peer reviewer, and just have that in the back of your mind. Don't automatically trust any paper that comes on your desk.
Of course.
Yes. Yeah, I think it's not their job, and they're actually not trained to detect manipulation. So those reviewers, they are just scientists. So there is no specific education to become a reviewer. And what I find interesting or critical is that they have time restrictions as well. They have their own ambitions and their own projects. So usually it depends on how advanced you are in your career. But when you have many reviews on your desk and you don't spend much time on them, and then you can't screen them so carefully. And some reviews, or maybe it's natural, sometimes you use easily available heuristics, such as, for instance, the significance of the results. And sometimes we hear from researchers that papers are rejected because the results didn't look nice enough. And so it's not their job. Yeah. Maybe one aspect that I would like to add is I think the role of peer review in data manipulation, it's not just that it doesn't detect manipulation bias or manipulation, that it not just can't detect it. I think it also creates incentives to manipulate your data, because often you anticipate that your paper won't be published when you don't produce nice results and you don't tell a nice story. And a colleague of mine, he's from the Netherlands as well, actually. He called it a good movie principle. So a paper have to tell a nice story, otherwise it's not interesting for the journal and it doesn't get cited in the end. And in our survey, for instance, we provided our respondents an abstract, an hypothetical abstract. And one part of our respondents got an abstract with significant results, and the other half got an abstract with significant, so significant with insignificant. And we asked them to rate the method and the contribution and the publication likelihood. And what we saw was a huge increase in the publication likelihood for the significant abstract. And afterwards, we asked them, why do you think it's more likely or less likely that this paper will be published? And 20% of the respondents somehow argued with significance. So 1 out of 5 mentioned significant as a reason for their judgment. And many of them actually said, I don't think that it will be published because journals don't like negative results. So there's
that's
two sides.
The stress of the peer review, I think, plays a role. The process itself plays a role in people trying to make their data look nicer. So they tend to play around the data and change things around, and that sometimes qualifies as manipulation. So the third question I have is, how often does data get manipulated, and how is it affecting other scientists and research?
Who can start?
It's a very difficult question because we can only talk about uncovered cases of misconduct or cases that researchers report. And it depends on whether you look on anecdotal evidence by analyzing, for instance, case studies, or analyzing retraction numbers and the retraction reasons. What you can also do is analyze empirical evidence by just asking researchers in surveys. But asking, I mean, I'm not sure how you would react if I would ask you whether you ever committed misconduct or whether you manipulated your data. It's a highly sensitive issue, and there are consequences when you admit to misconduct, and there might be consequences when you admit to misconduct in a survey. But still, we asked our respondents whether they actually manipulated their data before, and it's a very small number. So 1.3% of our respondents said, yes, I have manipulated my data before. So it's a very small figure, but we were actually surprised that at least one of them said, yes, I did that. And those figures are matched by other studies as well. Like, for instance, nowadays it's quite old studies, like 15 years ago, by Martinson, Scientists Behaving Badly. He found similar numbers, like 1 to 2%, but this is just a very lower threshold, a very low threshold. So I actually don't know, I would say. Maybe it's easier for image manipulation.
Yeah, for images. So I did a big survey of 20,000 papers, and I looked for image duplications. And now image duplication is not necessarily misconduct. It could just be an honest error. So we figured that if an image is just directly copied, it might be an honest error. But if an image is really manipulated, for example, you'll see the same cell 10 times in the same photo, that seems very likely to have been done intentionally. So using classifications like that, we estimated that about 2% of the papers with images had intentionally manipulated image duplications. And so that's just for images. So I think it's much easier to manipulate data in tables and line graphs. You can just make a beautiful line graph and put it out as a real data. For photo manipulation is actually pretty hard. You can sort of detect that. But manipulation in a table is much easier and much harder to detect. So I think the real percentage I would estimate is between 5 to 10%. And yeah, again, it's just a guess, but somewhat educated and somewhat based on data. And there are certain fields where I feel the manipulation is much higher. So in particular, small non-coding RNAs and oncology, that field, so like miRNAs and long non-coding RNAs, that field seems to have been heavily infected with paper mill papers that are presumably all fake. And we're finding by now thousands of papers that are completely fake and that are being produced by paper mills. And yes, they have infected certain fields. So I think in certain fields it's maybe up to 50%. And that's just yeah, a horrible number.
Oh my God.
I think it depends on where you draw the line, what manipulation actually is. And those mild forms, I think many researchers actually are not aware that this is already some form of manipulation and data tweaking, for instance.
Yes, and this
Right.
is affecting like the future research who bases their data on other papers. I think this plays a big role. And one more question: should more attention be given in education, training, and academia to the importance of scientific ethics and the use of data and the delivery of data? And how can we do that?
I think you can't possibly answer this question with no. So of course we have to train our students and our junior scientists, and I think this is maybe the job of universities and research institutes to train their junior scientists. And I would even go further and say that schools should somehow integrate some basic form of science education and how to evaluate whether a study has at least a minimal threshold of quality reached, for instance. And what we do in Zurich is training our students. So we offer courses on the scientific system, how it works, how the peer review process works, how you can distinguish a good and a bad study with respect to quality, or at least what are certain criteria, how you can evaluate studies. And of course, how to conduct better research. And of course, this has to be part of study programs as well, and PhD programs, and we have to teach students.
Right, and I'm going to add to that it's important that those classes also contain real life examples. I've seen some of them and they seem to be very dry, and I feel you really need to give students real examples. You know, you have these results and you have an outlier, what do you do with them? And not only teach students and young, you know, researchers, but also teach the older faculty, because I feel it's not like it's one thing to teach students what is right and what is wrong, but there are certain situations where you might end up in a lab where the professor or the group leader is a bully and demands that you give certain results. And I usually give the example in the United States, if you're on a visa and you're a postdoc and your professor is firing you, letting you go, then your visa also immediately is canceled and you have to go back to your country within a couple of days. So if you're in a situation like that and your professor tells you, I want your results by Friday and I want this PCR to work, then you're very tempted to do misconduct. And so it's also the atmosphere in a lab that determines whether or not science misconduct happens. And so you can start a lab and you can start your career with all the best intentions of the world. But I do feel the part of the problem is also with bullying group leaders who just create this atmosphere in the lab and the incentives to cheat.
And I don't know if in universities, like this is something this is why I asked the question. We have no clear idea about how these things work. I don't know if in universities the person has someone to go to or someone to ask in case he gets this kind of bullying or this kind of request from his instructor. Is there any possibility in universities, or what is usually the process?
I would say that most universities have ombudspersons, so persons you can talk to, and most universities, at least the ones I'm familiar with in the United States, will have also an Office of Research Integrity, so you can go to them as well. But unfortunately, it's the case that when it's the word of a young researcher against the word of an established professor, unfortunately, it's the professor who will be more protected by the university, and it's the young researcher who might lose their job, even if they're completely right. And this is completely unfair, but yeah, so it's, you know, you can have beautiful structures in place, but in reality, these misconduct cases are often swept under the rug, ignored by and not addressed by universities. because the professors are, you know, the established people who bring in the money. Usually they write the grants and bring in the money for the universities. And so universities don't often want to address these problems. So a very unfortunate situation. And I hope that's going to change, but it will take a lot of time.
Yes. And the last question from me, and then I will be going to the questions from the audience. What is the role of the scientist, and what is your recommendation for this problem? What is the role of all the scientists, the scientific community, and what are your recommendations for people now going into science and or established scientists when it comes to misconduct and the manipulation of statistics and images?
Difficult again. I think we should all be aware that we have to commit to research integrity and to research ethics, and to educate ourselves and to always question ourselves whether we are still in line with research ethics and whether we still conduct research for the right reasons. I mean, it's very difficult, and what Elisabeth just said, it's true. I mean, it's very difficult for junior researchers to speak up. But it's very important that we try to, and I think most PhD students will come at a point where they think about quitting. At least I did reach this point during my career, where I thought maybe I should just stop, because it's not fun anymore if you have to compete with senior scientists and so on. But otherwise, you can't change anything if you drop out of science and if you leave science. So everyone committed to research integrity should try to make it better in the future and to treat others better than they have been treated, for instance. Yes, but there also need to be institutions that somehow make sure that research integrity exists and or is spread. Like we have to, for instance, we have to increase the incentives to publish negative results. We have to increase the detection likelihood of scientific misconduct. We have to keep looking for misconduct, and we have to sanction it. I mean, it's a very clear social concept. If we don't sanction, then there's no reason not to do it in the future. So we need sanctions and detection methods and so on.
Yeah, I completely agree with all of that, Julia. That is great points. I feel we need to have more people who are committed to fight misconduct. So I sort of am at a more senior point in my career. So I quit my job, and this is now my full-time job, unpaid most of the time. And I'm calling out attention to misconduct. I give a lot of examples on Twitter, make it sort of in a fun, almost a game type of way, as a puzzles. But I'm not alone. Like, there's many people who are like me, and they're usually fighting behind the scenes under pseudonyms, because it is very hard to find misconduct when you're still employed. There's all these types of conflicts, and probably your employer is going to tell you that you cannot tackle certain problems because you're being seen as a nuisance, as a problem. And so I felt that for me, the only way to do what I'm doing right is to quit my job and to not be attached to any university. But I'm also in a fortunate situation where I can afford to, work a couple of years for free, do some consulting, which is what most people cannot do. But I do feel that the work that we have been doing, that it's starting to yield results. That, for example, journals are paying much more attention now to images and to also other things like conflict of interest, animal ethics. That was something that some journals didn't pay attention to, and that's starting to change. There's a change in the past couple of years in whether or not the journal will retract a paper based on image manipulation. A lot of these cases were swept under the rug again, were ignored, but I'm calling them out. I'm calling out the journals on Twitter and trying to get papers retracted where I just feel that, the images have been manipulated so much. And I feel we're getting more successful in those things. So I do feel there's a change for the better. But, scientific publishing and science as a whole is just such a slow and old process. It's just gonna, you know, it's not gonna be a sailboat that swiftly goes to the other side. It's like a big oil tanker that takes, you know, hours to turn its direction. And so, yeah, it's going to happen at the glacial speed, but it's I'm optimistic that we're getting better as a whole. But yeah, we need to, it's not a problem I can tackle alone. Like, we need many more people, you know, putting their effort in it.
Yes, of course. And as a follow-up to what you're saying, one of the audience asked that some journals now are asking for the raw data for the images. Is that true?
Yes, some journals have already asked for that for a long time. Unfortunately, I've seen some beautiful image manipulation in the raw blots where they just like make, I think they took an old blot and they just superimposed, photoshopped some bands on top of that and made it look like it was a, you know, had old scratches and things on it, but it was all manipulated. So I do feel it's definitely a good thing, but yeah, it's also, you then also need to pay attention to how the raw data looks like, and not just, you know, check the box if a file has been given, like actually look at it, because there's image manipulation going on there as well.
Okay. And if I may add, a lot of journals not just request the data, they also request any code and do files or R program scripts to actually replicate the data, and like a README file where you explain how you can actually replicate the results on the paper, which is in fact a criteria of quality, I would say, even though you don't have to actually run any analysis again, but just having the data there, for me, it is a signal of a quality of a paper if it provides the data and the code to reproduce the results.
Exactly, yeah, like the willingness to share your data.
Yeah.
That's already, you know, gives so much trust in the data. While in a lot of the papers I'm dealing with, where I suspect manipulation of the data, the authors are always very hesitant, and they'll give a new set of photos, for example, but they never actually share any data, any, you know, like for example, the 3 data points that made the mean value. Like, if you cannot share that means you don't have it, and you just made up a graph.
Yes, exactly. And in my most recent publication, I was asked to give the calculations that I used to get the significance and to get the images. Like, they're asking a lot of questions before I can send my images, and the Excel sheets should not be as images. And I think that's on the good track, moving on the good track. So now I'm gonna roll the first poll. Actually, there are 2 polls. They're gonna be running at the same time, and I'm gonna wait for like a few seconds for people to vote, and we're gonna go over the answers. So the first one is, do you have second thoughts when citing results or basing your future ideas on them because you fear data manipulation? And do you usually feel more confident when looking at results from high impact or well-known journals? And I think like these questions are mostly from me, because I always think about the data that I'm seeing and I'm replicating, because I've seen like citations from papers and that are not, I see the sentence, I go to the original paper to get the full image, and I don't even see the data that they're citing, and it's not a good thing to do. And the second one, like, I feel more confident when looking at a high impact journal. Personally, I feel like the peer review process may be more thorough and they look more into the details. So I guess that's only my opinion. So we're gonna see the others, what they think. So we still have a few people to vote.
I think we can share the results. I think the last few are shy to vote.
Okay. So I'm gonna share, okay. So most of people haven't thought about data manipulation or thinking about seeing the results, or if the results were manipulated before citing them. And most people feel more confident and more comfortable working with results from high impact journals. So I don't know if you have any comments about this, Drs. Elisabeth and Julia, if this is something that you've seen before or you've heard about it before.
I think with respect to the last question, I think it's a common misperception that high impact factor somehow is related to quality. I mean, the impact factor, it's not a quality measure, it's just a citation measure. So a high impact factor just means that research published within this specific journal is just more cited than the research published in other journals. For me, what is important is whether they have an actual review system and whether they actually have reviewers that invest time in doing the reviews. And so I wouldn't put too much emphasis on the impact factor of a journal. And I think it's not necessary to consider the impact factor if you just very carefully read the paper, and if you form your own opinion on the quality of a paper, then you don't need such criteria such as citation number or impact factor. And clear criteria for me is how transparent they, for instance, report the results, whether the data is available, whether they have made very clear why they conducted their research in this specific way, whether they discussed alternative explanations or limitations and so on. So I think I actually forgot what I clicked, but I think I clicked, I don't look at the impact factor usually when I cite research or when I have a look on research. And I forgot the first question. Maybe Elisabeth wants to answer that one.
The one about impact factor?
Yes, or the first one. I forgot what the first one.
Okay, there it is. So I feel I have some experience, of course, with data manipulation and finding those papers. So I feel I have some idea whether or not I can trust the paper. I might be completely wrong again, but I probably have a little bit more experience. I've seen so many papers. So I feel when I cite a paper, I have a pretty good idea if I can trust it, just based on, I usually don't look too much at citation index, but like they might be a group of people who have published a lot on that particular field. And so I trust their results, and, you know, I have maybe even replicated their results. So I do fear data manipulation a little bit, but I usually, yeah, have a good feeling of whether or not I can trust the data. For the second question, I did find in my research, so I did 20,000 papers and I looked at different journals. So I looked at 40 different journals, and there was a negative correlation between impact factor and the amount of duplicated images. And it was not perfect, but yeah, the higher the impact factor, like Science and Nature, I found fewer of those duplicated images than in low impact journals. But there were some low impact journals that actually did really well. So there were, it might be also dependent on the editor-in-chief and the team that is behind the journal that looks for the, you know, quality data. But I did also find image manipulation or image duplication in Science and Nature as well. And so there, it's you cannot trust it completely, but there seemed to be a little bit of a correlation between the two. Yes.
Okay, thank you.
That's just looking at images, so I don't really know about other data, but looking at images, yes.
Okay, thank you so much. There are people who are sharing books about how to lie with statistics, and some really nice books if you want to check them out.
How to lie.
Yeah. Okay, so now I'm gonna ask people if they want themselves. So we have a list of questions. I recorded them all. I can ask them, but I want to see if people want to ask themselves their questions to explain what they think. So does anyone want to ask themselves the question?
I'm happy to go ahead.
Yes.
Julia, I'd like to ask you for your opinion on pre-registration. So I'm in biology, where pre-registration is not that common, but from my understanding, pre-registration would be a really good tool in minimizing publication bias, because people commit to a certain analysis in advance and they commit to a certain data that they will publish. But since you have more experience in this field, I was wondering what you think of pre-registration.
It's an interesting question, and I think important is to distinguish publication bias and manipulation. And I feel like pre-registration, of course, it's a great tool to avoid manipulation, because the study is very clearly checked, and you commit to a plan. You have to write down in advance what you're going to analyze, what your hypothesis is, what you expect, how you're going to analyze it. But publication bias is on a publication level, so it's not. So pre-registering your study doesn't might not decrease publication bias, because it's still researchers have to decide whether they write their results down, and journals still have to decide whether they want to publish it. And there are some studies, and I found them very intriguing and interesting, where they followed a group of pre-registered studies and just checked whether they have been published later or not. And what they found is that of all those pre-registered studies, the studies that had significant results were much more likely to be published. And the studies that hadn't found any nice results, so to say, they were much less likely to be published. And then this is actually publication bias, not writing down results that don't tell a good story. So pre-registration might reduce manipulation, but not publication bias, I would say.
Thank you.
Thank you. Is there anyone else that wants to ask a question to our speakers?
So I'll probably ask. So, hello. So I'd like to ask, so you know how in a study they can show the data they want to show and just hide whatever they don't want to show you. And for some types of data, like for example, a Western blot, you can see whether they have cut the blot and things like that. But for example, for I don't know, counting cells in brain tissue, they're not going to show you all the slides of brain tissue that they have been counting on. So how do people verify this kind of data? Is there any humanly way to verify this?
Yeah, I mean, when I just think about sequencing data, when you have like uploaded 2 million reads, I don't think there's any peer reviewer who will download that and to check if the results are there. Like as a peer reviewer, I would not even know how to download such big data sets and analyze them. So I usually hope I'll check if the data is available. If it is, I feel that's, you know, good enough. And but yeah, it's very tough because there's so much data hidden that is not shown in most papers. Like usually, even if you're supposed to share the raw data, I feel like when you just present a bar graph with an error bar, like in most cases, a journal would not ask for the raw data there. Like what are your measurements there? And yeah, that's there's no way of checking all of that. And a peer reviewer is not going to check it. But I do feel like what Julia said, when authors are sharing data, it is a sign of goodwill, and not that I think that most people will download it. Maybe 1 or 2 researchers who are interested in the data will actually work with the data, but I don't feel a peer reviewer will look at it. But just knowing that the data is available is a big plus.
I think it's not always about checking what is not there. Sometimes it's enough to check what you have. And there are some case studies, and I found them very interesting, where researchers found manipulation by checking what is there. And they, or I think it was one guy, Uri Simonson, he had a look on the data and he realized this data is too good to be true. So the results were so extremely perfect. So he had like 16 hypotheses, and all of them were perfectly matched by the data and in very strange ways. And so he, for instance, he conducted some simulation studies and found that the result that they published was super, super, super unlikely. And he requested original data, and he confirmed his hypothesis that the data was manipulated. I think the reason for that is that when researchers fake their data, humans are quite bad at faking data in a good way, because we have a wrong understanding of what chance is actually. And if you fake a data set, it's actually not a random sample. And if you're very sophisticated in statistics and have a good intuition, then you might see that your data is not random. And I think this might be the same with Western blots and such things. You always have this white noise inside, and you can't reproduce noise in your data, and you can't manipulate or fake noise in data. So sometimes it's helpful to just have a look on the patterns that you see, whether they are realistic or not.
Okay, thank you so much. And I'm gonna give the floor also if someone wants to ask a question. You can raise your hand, by the way, if you want to ask something, or just go ahead. If not, I'm gonna start asking the questions that are written in the chat box.
Sorry, I would just like to ask on what Julia said, because you say you can't fake white noise, but is it? I don't know if I remember correctly, but Elisabeth, I think I've seen some of your publications on Twitter where the people have faked white noise in Western blots or other publications, haven't they?
Yeah, I mean, we've seen, we caught this paper mill, and like I wasn't the only one working on this. It was a group effort, lots of other people noticing the same thing, where we had this group of Western blots that all had the same background. So it had some noise, but they all had the same noise in all the same panels and all the same figures in that paper, but also across 500 different papers from 500 different universities. And so they faked the noise, but it was just, they made the mistake of inserting the same background noise all the time. So we think this was an artificially generated blot program, but by accident, they didn't add, didn't add any noise to the noise. So the noise was always the same. I'm sure that because we caught this paper mill, this group of papers that all had that same background, that now the design studio has added a little bit more noise to it, and now we cannot catch them because there's no duplication. We can catch duplications, but we cannot really see if a blot is fake or not. So it's it's a two-edged sword, where in one time we're catching the bad guys, but we're also teaching them how to be even worse at or better at faking. And so it's it's like if you have a good noise generator for images, but also for data, like it's very hard to catch that. We're really only catching the very dumb cheaters, I feel. And so that is it's probably just the tip of the iceberg. So that's a scary thought.
Okay, we have someone raising their hand. Go ahead.
Hi. It's a question about the reason behind this behavior. We talk about the ethical aspect of this behavior, but don't you think that also the publication system itself is the cause? Because the impact factor, the journals, and then behind it is the grant also that make people doing such thing. And in the other hand, even the system itself, for example, the reviewer are not paid. So who is going to check? To thoroughly check this kind of data? Or it's not, for sure, it's not the reviewer, because it's waste of time. He's not getting paid as well. So
yeah,
that's
a great question, and the incentives for misconduct are really high. Like, we all feel the pressure to publish. We all want to publish lovely, beautiful results. And so there's a lot of pressure to fake results and to do misconduct. And there's very few punishments. Like one of my personal frustration is just the lack of retractions and the lack of punishment for researchers who have been caught. And they're often protected by their universities. So people can keep on faking results because they're not being punished. And so I think we're showing the world, and that's okay to cheat, because you'll still have your job and you'll still be able to do research. And that is not how it should be. Like if we could always run red lights and be earlier at our work or where we need to be, then we would all massively run the red lights. But if we just know we can be caught and get a ticket for running a red light, then we're a little bit more cautious. It's like all of us in a way want to cheat, but we know that we will be punished if we do. And if we notice that there's no punishment, more and more people are going to cheat. So there need to be better punishments and better, just taking away jobs or taking away your rights to apply for grants. Things like that could be good punishments for people who have been caught cheating, but that's not happening.
Okay, so I don't know if anyone also wants to ask a question. So I'm gonna go ahead and ask a question from the audience. What are your thoughts on pooling data from an experiment conducted on different days but maintain the same conditions? I'm conflicted about conducting statistical tests to determine variability, and any guidance would help.
Oh, that would depend on the type of experiment. Like I would say measure, let's say, the light emission from a light bulb, that probably is the same on if you measure it on Wednesday or Thursday. But anything with biology, there could be a huge difference. I don't know, some cells just only want to grow on Wednesdays, and we've all dealt with that. Like, my PCR only works if I give a little magical curse, and then it's going to work. I feel that in biology, there's much more variability that we cannot really explain. And it might be dependent, for example, on whether if a lab is cleaned once a week or so, maybe, your cells or a PCR are going to work better on a particular day of the week. And there's just hard to predict which things are of influence. But so usually when I did, for example, DNA extractions, I would try to do mixtures of extraction. So I would all extract different types of samples on the same day, and not only extract every sample from one patient on one day and then the next on another day, because I just felt that any DNA extraction had its own variability. So it's better to mix samples a little bit in these DNA extraction runs. But it's tough. It's you would first need to prove that there's no effect of day of your experiment in the results.
I'm not an expert in that field, but isn't this easy to just control for the day in your statistical model? So if you include the day and the time or whatsoever, you might control for that effect or not.
Yeah, yes. Yeah, I mean, it's hard to answer a question without knowing a lot more details.
Yeah, sure. but from my social scientist perspective, if we have such cases where we have different conditions or different days and times, we just control for them. But on the other hand, we believe by controlling for variables, we find causality, which is not true. So just controlling for everything is not a solution, but I feel like it might be possible, maybe.
Okay, so thank you. All the other questions are things that already have been almost answered. I'm just reading a bit about the questions. They are mostly answered before, so I will not ask them again. Is there anyone that wants to ask any more questions? Because we have 10 more minutes. Yes, go ahead.
Hey, oh, sorry.
It's okay.
Am I talking over someone?
Go ahead. Yes.
I was wondering about incentivizing journals to hire editors specifically to look at statistics and data. It seems to me there is no incentive for them to spend money to do this. And they seem to be the gatekeepers, so they should do it, but how can we get them to acknowledge that they are the gatekeepers in this process and that they have a responsibility to check rigor?
I mean, it's difficult since they have a monopoly, and the peer review, it's a barrier. It's not just a quality control, it's also a barrier to journals. But I think one incentive might be that the number of retractions will decrease if they check properly in the beginning. And I mean, retractions, having a lot of retractions, this is a decrease in reputation, I would say. And if you don't have proper mechanism against manipulation in your journal, then you will have a lot of, you might have more retractions than other journals, for instance, and this will decrease the reputation. And I think Elisabeth, she said it before that I think you found less image manipulation in high imperfect journals, but on the other side, they have more retractions usually. So the number of retractions is higher for high imperfect journals, and I would say this is a loss of reputation. But
how can we make that reputation? I mean, if they're still high impact, almost we would need another metric that then, you know, reflects that loss of reputation. Yeah, I don't know. I agree.
No, very good question. I actually have no idea how to make them see that it's a lack of reputation.
Yeah.
No, but I do think that there's a slow change that I've been seeing in journals where they start to care more about it. I'm not quite sure about hiring statistical experts, but definitely for image manipulation, ethical concerns, they're doing more checks, because in the end, it's much less work for a journal to do this work up front before a paper gets published than to do it afterwards. And again, this is a slow change, but yeah, I feel as data sets are getting more and more complex, as a peer reviewer, I don't always have the capacity to know particular parts of a paper. So not just the statistical parts, but papers get so complex that I feel that a peer reviewer could see one part of a study and peer review that really well. And so there's some role in editors to assign different types of peer reviewers so that each of them can look at a different type of the study, a different aspect. But often statistical knowledge is the one that is lacking with both reviewers. And then how do you make sure that that is covered? So I usually will write to the editor, like, I cannot judge the statistical methods here. You need to have an expert. And some of the journals have a tick box where you can say some additional statistical consult is required, and they do that then also. So I do think there's a change in the way that journals are adding those additional checkpoints and even hiring people to do those checks. But it's very slow. You might not notice it yet.
Okay, so one last question. Do people assess whether or not a given journal is a predatory journal that publishes anything? And if so, how do we do that?
It's a tough question. A lot of people have tried to make lists of predatory journals, and it's tough because a lot of journals are in that gray factor. Like they seem legit, but they publish anything you send to them, and it's just very dependent on the editors. And there have been, in particular, independent journals that are not associated with one of the bigger publishers. There's a couple of rogue journals that are indexed in PubMed and they just publish whatever is sent their way, and they have like complex structures in which the editor only publishes in their own journal. And that even actually happens in some journals that are associated with the big publishers. And so it's not always clear what a predatory journal is and what is not. There's some very clear examples on both sides, on predatory and legit, but there's just a huge gray zone and not something that is easy to answer. In the end, most publishers care more about money, I feel, than about science. So in the end, you could argue that any journal is a predatory one.
Often they have very nice names as well. So they sound like traditional journals in their discipline. So it's very hard to identify them. What might help is have a look on studies that have been published there and form your own opinion on whether you think there is some quality in it or not, and whether the data is available, for instance. And I think the problem is also that many people rely on the impact factor on where journals are listed, but there's also the period before a journal gets assigned an impact factor, for instance. So if you start a small independent journal in the beginning, you don't have an impact factor. So it takes a few years until you get one. But I feel like I get a lot of invitations of very strange journals, and then the spelling is strange, and you have a look on the website and you actually see that this is not an actual journal. I mean, or maybe not a real scientific journal. And I think it might help to carefully inspect the website. And once you are asked to pay a fee, you have to carefully check whether it's reasonable or not, whether you get peer review or not. And usually predatory journals, they don't have peer review.
Yes.
Or they have a fake peer review where they just check the spelling or whatever.
Right.
Yes,
I mean,
yeah, sorry, go ahead.
No, go ahead.
No, I sometimes get Twitter messages from journals like, do you want to publish? Do you want to publish? Like, no, I don't want to publish. I don't have anything to publish now.
They leave me alone because they know that I'll, immediately if they send me an invitation on Twitter, I'll make them ridiculous. So they'll think they leave me alone now. But I mean, yeah, we all get those, like, dear esteemed researcher, we would love your contribution. But some of those are actually from real journals. It's hard to tell them apart. Yeah. For me, I think I usually check, are they indexed in PubMed? Because if I want to publish my paper, I want other research to see it. If it's just a standalone journal and it's not indexed in PubMed, yeah, none of my coworkers are going to see that paper. So that would be, it's not an easy or like a foolproof method, but that's one thing you could check. Is it going to end up in the index that I use for my literature search? And if not, then that might not be a journal I want to publish in.
Yes, exactly. And now we're running out of time. So I want to thank everyone for attending. I want to thank Dr. Elisabeth and Dr. Julia for taking the time to participate with us. I don't know if you feel comfortable in me sharing your Twitter account for everyone to follow you. Is this okay?
Oh, yeah, sure.
Yeah, sure.
Okay, so I'm gonna
put it. Oh, okay.
Yeah. And sorry if I missed any of the questions because we're short on time. And make sure to follow us on Twitter and create a Scientistt account where the community is very helpful. We always have events running, and we have 2 more events running in March. So check them out. And thank you so much for everyone for taking part of this, and have a nice evening, or have a nice day if you're if you're in the US now.
I'm just starting, yes.
Yeah. Have a nice day, Dr. Elisabeth, and have a nice evening, Dr. Julia.
Thank you very much. You as well.
Thank you, Jennifer and Hassan, for inviting us. It was my pleasure to be here.
Thank you for such an interesting topic. Yeah. Have a good weekend, everyone.
Thank you.
Bye-bye.
Bye.